This is video lecture 4-2, Population Ecology. In video lecture 4-1, we introduce some basic concepts in ecology, including thinking about the factors that influence where a species can be found. In this video lecture, we are going to talk about how a population might respond to a particular set of factors in the area where it is found. Is the population growing, shrinking, or stable in size? How do we measure that? And if the population is growing, how big can it become? What limits the ultimate size of the population, called the carrying capacity? These issues are of concern to ecologists and also to conservation biologists interested in understanding what the potential fate of rare and endangered species might be. For example, originally there were 15 to 100 million bison on the Great Plains. Hunting decimated the bison population, reducing it to only 1,000 buffalo in 1889. Conservationists began establishing protected herds of bison in the early 1900s. In Yellowstone National Park, the protected herd started with a population size of only 21 bison. So monitoring how the population changed each year was important to determine whether that herd would grow in size and recover, or whether that herd would decline in size and go extinct. As you can see from the data here, with year on the x-axis and number of bison on the y-axis, that the herd increased in size and recovered, and in fact today there are lots of bison in the park. We also want to manage populations of organisms that we use as resources. We harvest a variety of organisms for food, such as lobster and salmon, and many more terrestrial and aquatic plants and animals. Obviously, you need to be careful when you harvest individuals because you don't want to leave too few so that the population goes extinct. But in order to know how many to take, you need to know what the population size is every year and whether it is growing or declining. If the population is declining, you can't take any without driving it to extinction. But if the population is growing, you can predict the size of the population after accounting for how many individuals you're going to harvest to make sure you're not causing the population to get smaller. The first step in understanding populations is to estimate their size. This can be done by a few different methods. One method is to use something called quadrats. With quadrats, you count the number of individuals in a specified area to estimate density, and then these can be used to estimate population size. You need to set up the size of a plot based on the size of the organism. So for instance, in these plots, the organisms being counted are relatively small. Think about if you were interested in a population of trees, however. You would have to set up a much bigger plot. Using quadrats to estimate population size is particularly good for plants and fungus and other organisms that don't move, or for getting an instantaneous count of the population. Another method is called mark and recapture, and this is good for organisms that move. The idea is that you capture a number of individuals and you mark them all in some way. To mark organisms, you can use tags or place paint on the outside of the organisms, like on these ants or the crab. In addition to these sorts of external tags, you can insert pit tags into organisms, like you might have an ID chip implanted into your pet. These pit tags provide a barcode that can be read using a barcode reader. Finally, you can use natural markings to identify individuals that have unique markings, such as this red-spotted newt. The pattern of red spots on their heads and back are unique identifiers. After you mark the organisms, you release them back into the environment, and at a later time point, come back and recapture a bunch of the same population. You then count up the number that you recaptured that are marked. We're going to talk about this method of estimating population size in more detail during class. Okay, we can measure population size, but what we might really be interested in is how the population size is changing over time and/or how population growth differs, either across time or between populations in different sites. So how do we measure the change in population size? We use the number of births, represented here by a B, and the number of deaths, represented by a D, to estimate a population's per capita growth rate, or the change in population size per individual per unit of time. Again, you would use a time period appropriate for the organism of study, years for humans, but maybe days for bacteria. We are going to work with this equation and the others that follow it in this lecture during our class time, but think about these situations. Sometimes the number of births in a population is greater than the number of deaths, and sometimes the number of births is less than the number of deaths. What happens to the population in these two instances? The value R is an important measure because it tells us, first of all, whether a population is increasing, stable, or declining, just by looking at whether it's positive, negative, or zero. And its actual value will tell you how fast a population is growing or declining. As R gets more positive, population size increases faster. As R gets more negative, population size declines faster. So let's talk a bit further about the value of R. If you have an R of 0.5, by definition, it means your population is increasing by 0.5 individuals per unit time. What does that even mean? I'll tell you one thing. It does not mean that your population is increasing by 0.5 individuals per year. But wouldn't it be easier to understand changes in population size if we knew how many individuals were being added or subtracted to the population each year? If R equals 0.5, how many actual individuals are added to the population? Well, that's going to depend on what your population size is. In order to determine how many individuals you add or subtract, you need to multiply R by the population size, represented by N. Population growth is an important measure in understanding what is happening in populations. In this example, let's look at whooping cranes, the largest bird in North America. Before European settlement of North America, the population number may have been as high as 10,000. Conversion of prairie to farmland and hunting drastically reduced their numbers. By 1944, only 21 individuals were left in one population. With the creation of the National Wildlife Refuge in Texas that protected its winter habitat, populations of whooping cranes started to increase. This is an example of exponential growth due to recovery from a very small population size. Here's another example, the introduction of sheep to Tasmania. In the early 1800s, farmers brought sheep over. They had unlimited resources, so their populations expanded greatly. But look at the graph of the growth in population over time. Did the population continue to grow exponentially forever? No, and the limits to population growth are universal because there are only so many resources on Earth. Population growth is limited by the amount of resources that population has. So what does limit population size? Think about the sheep in Tasmania. Tasmania is an island, and so the amount of land and grass cover is finite. There's only enough room and food for a certain number of sheep. This is an example of a density-dependent factor. Density-dependent factors increase as the size of the population increases. Some other examples are nesting sites, competition for food, or competition for water. Can you think of others? Other factors that limit or affect population growth are not dependent on the number of individuals in the population. For example, temperature will affect individuals in the population regardless of how many individuals there are. Typically, both types of factors are in play and combine to affect the potential population size at any given time. As we said before, temperature affects winter survival of sawflies, and warmer winter temperatures may mean that the population is higher in the next year because more overwintering adults survive. However, in the summer, the population is going to be affected by parasitic wasps that cause larvae to die. The higher the population of sawfly larvae, the more successful the parasitic wasp, and you get a density-dependent cycle. Mussels are typically limited by density-dependent factor of competition for settling sites, but sometimes disturbances will remove mussels and allow for increased population growth. The combined factors acting on a population result in an upper limit to the number of individuals that a population can sustain in that particular environment. This limit is called the carrying capacity. Carrying capacities are specific to a location and for a species. And carrying capacities can change when the environment changes. For example, if there is a change in temperature or a change in the interacting species, such as predators or parasites in that particular environment. Imagine a population with 2 individuals. At first, there are plenty of resources for everyone, so the population growth is rapid. But as more and more individuals inhabit their habitat, some resources begin to dwindle. Competition for resources increases so that birth rates decrease or death rates increase, and population growth slows. Eventually, the population reaches a point where the birth rate is equal to the death rate, and populations cannot grow anymore because there are not enough resources to support more individuals. This growth pattern is represented by an S-curve, or logistic growth curve. For organisms we use as food or fuel, we want to minimally affect growth, so we need to determine how many individuals we can harvest and minimally affect population growth. The point at which we minimally affect population growth is that point where the population grows the fastest. Because it's growing the fastest, it can recover the quickest at this point. The aim is to maintain the population at that point of maximum growth, so we want to maintain population size of K, carrying capacity, divided by 2, which is the point of maximum growth. You can sustainably harvest the amount of individuals it would take to decrease the population size to K over 2. At a population size of K over 2, there are enough individuals that finding a mate isn't a problem, but not too many that overcrowding and competition is a huge problem. This is called the maximal sustainable yield. But how do we know what the carrying capacity is? Do we wait for a population to stabilize in growth and then begin to harvest? Well, that would take a long time. We also often don't know how many individuals are living. We don't have good data of populations, especially for fish that live under the water. Finally, maximal sustainable yield doesn't tell us which fish to catch, young versus breeders versus old. If our entire harvest consists of breeding fish, we might actually cause populations to decline because we removed individuals that give rise to more fish and the population wouldn't be able to regenerate. There are issues with implementing this strategy. This is New Zealand's highest value fishery. These fish are called slimeheads because they have mucus pores on their heads. But the name was changed to orange roughy to better market them for food. Who would want to eat a fish called a slimehead? This species lives in deep waters off the coast of New Zealand, which has made research on the fish difficult. Many northern hemisphere fish have maximum sustainable yield, and that was applied to orange roughy too. This was a huge mistake because it turns out that these guys live for up to 149 years. And you know that for an organism with a long lifespan, there's a trade-off with age at maturity. Longer lifespans mean later age at which they become reproductive. The orange roughy doesn't start breeding until they are 30 years old, which means that if you take 70% of the population, there are not enough fish that are older than 30 to reproduce and maintain the population size. Remember, we'll be working through more of these equations and concepts in class.